Let us continue our discussion on adaptive immunity, otherwise known as acquired immunity. Just quick review, we have two types of immunity. We have innate, which we're born with and is non-specific to any pathogen, whereas acquired or adaptive immunity is very specific to a pathogen, whether it's varicella, chickenpox, smallpox, Ebola, Corona-19. So we talked about pathogens, and pathogens are something foreign entering the body that is bad for our body, that may cause disease, sickness, or illness. And pathogens have cell surface markers made of proteins, so protein surface markers known as antigens. And it is a bit of a misconception when people hear the word antigen that it's something foreign or even something bad. It's certainly something our immune system focuses on and identifies, but we have endogenous antigens and foreign antigens. Endogenous meaning they're within our body, and they identify our cells as self. That is to say, all of our cells recognize each other, or more specifically, our immune system recognizes all of our body cells because our body cells have antigens that identify themselves as belonging to that specific body. As autoimmune diseases are problematic because the immune system is no longer able to identify self with certain tissues in the body. So antigens are nothing more than cell surface markers, either on our body cells or on foreign organisms or pathogens. So like we have here with Tim has an identification tag identifying who he is. And there might be other individuals that look very similar to Tim, but they have a different identification tag or antigen, if you will. It's denoting who these individuals are. We have Sally, we have Fred. But now if we look at Tim again, Tim is looking at these other individuals because he realizes they don't belong to him. They don't have the same antigens as he does. So the Sally antigen, the Fred antigen, and the Joe antigen would be analogous to foreign antigens or exogenous antigens that don't belong with Tim. So lymphocytes, both T lymphocytes or B lymphocytes, which are known as T cells and B cells respectively, focus on foreign antigens. Antigens that are bound to foreign cells, organisms, or viruses that enter our body. Let's just take a look at how lymphocytes divide and become activated. Now I have T lymphocytes shown here, and the same idea works with B cells, which we'll talk about. But lymphocytes will divide into memory cells and effector cells. So once a lymphocyte becomes adaptive or recognizes foreign pathogens, then those lymphocytes exist for the individual's lifetime or for nearly the individual's lifetime. But we want to always have a clonal population of those lymphocytes ready to be mobilized in case there's an infection. So lymphocytes will divide into two different populations. They're going to divide into a population known as effector cells, are the ones that are going to carry out the activity of that specific lymphocyte. And then we have memory cells. And memory cells are not carrying out any activity. They are just being sequestered and saved for a later date or upon further exposure down the road. If there's further exposure down the road, say 1, 2, 10 years down the road, we will have memory cells that identify that specific pathogen based on the pathogen's specific antigens or cell surface proteins. And the memory cell will recognize that. The memory cell will start dividing into effector cells and more memory cells. The whole idea of memory cells is the effector cells are eventually going to die off, and we want to make sure we have cells that are identifying the virus or bacteria. So upon subsequent exposure, there's a much quicker immune response. It takes quite a while for the immune response to be developed on the initial exposure, but subsequent exposure, because of the memory cells, allows for the immune system to act very quickly and rid the body of that bacteria or virus, oftentimes before the individual even gets sick. So next video, we'll talk about antibodies and B cells.